So, hello and welcome everyone. I am Beth Mashesky, and I'm the Environmental and Social Systems Research Lead here at the Illinois Sustainable Technology Center. That's part of the Prairie Research Institute at the University of Illinois. As a land-grant institution, the University of Illinois at Urbana-Champaign has a responsibility to acknowledge the historical context in which it exists. In order to remind ourselves and our community, we will begin this event with the following statement. We are currently on the lands of the Peoria, Kaskaskia, Piankashaw, Waya, Miami, Mascoutin, Odawa, Sauk, Miskaki, Kickapoo, Potawatomi, Ojibwe, and Chickasaw nations. It is necessary for us to acknowledge these Native nations and for us to work with them as we move forward as an institution. Over the next 150 years, we will be a vibrant community, inclusive of all our differences, with Native peoples at the core of our efforts. This webinar and all of the Illinois Sustainable Technology Center's webinars are certified green events through the University of Illinois Institute for Sustainability, Energy, and Environment. To find out more about certified green events through the University of Illinois, please visit sustainability.illinois.edu. To find out more about the Illinois Sustainable Technology Center's webinars or to sign up for our events email list, please visit istc.illinois.edu/events. Some housekeeping items before we get started. The webinar is being recorded. We'll send out an email announcement to everyone who registered for the webinar once it's available. Everyone will remain muted for the entire webinar, and we'll be holding questions until the end. During the webinar, you can submit questions at any time through the Zoom Q&A feature. You will be able to upvote the questions within the Zoom Q&A feature using the thumbs up button under the question. And then at the end, when we take questions, the most popular question, if any were upvoted, will be asked first. And then after that, in the order in which they were received within the Zoom Q&A feature. If you are interested in asking your question verbally, please raise your virtual hand and I will change your permissions to allow you to unmute. And then, of course, if you have any technical difficulties, please send me a private chat message. So with that, I'm very pleased to welcome today's speaker, Bryan Berger. He is a professor in the chemical engineering department at the University of Virginia. He earned his bachelor's degree in chemical engineering from the University of Illinois at Urbana-Champaign and earned his PhD in chemical engineering from the University of Delaware and was an NIH postdoctoral fellow in biochemistry and biophysics at the University of Pennsylvania. Bryan's research lab at the University of Virginia uses biotechnology to solve biomedical, environmental, and industrial challenges and seeks opportunities to translate these technologies to market. Their work is highly interdisciplinary, involving close collaborations with clinical, government, and industry partners. Bryan's work has been recognized by the University of Virginia with the 2022 All-University Office of Research Award in Public Impact Research and the 2025 School of Engineering and Applied Sciences Innovation of the Year Award. Research from his lab has been featured in major news outlets such as the New York Times. Additionally, Bryan serves as the associate editor for the Frontiers in Synthetic Biology. And Bryan is also the founder and CSO of Lighthouse Technologies, which develops novel biosolutions to address the major challenges in human and planetary health. So thank you, Bryan, for joining us, and the webinar is yours.
Thanks. Yeah, I appreciate it. It's nice to have the opportunity. So today I'd like to share some research from our lab broadly in the area of biotechnology for sustainability, and with particular emphasis on applications in food and agriculture. Thanks for the kind introduction. Our lab is really focused on the use of biotechnology to solve problems, whether they're human health, environment related, or increasingly focusing on biomanufacturing as a platform to make these things scalable. And we're always very excited about opportunities to work with industry, collaborate, external partners to see how these solutions that we're developing can have impact. My lab is I'd say these are sort of the main themes today. I'd like to focus on two areas. First, talk a little bit about work that we're doing in addressing environmental antimicrobial resistance in agriculture. We also work on biomineralization, looking at critical minerals recovery, brought biomanufacturing more broadly as a platform to make sustainable materials. And then at the end of the talk today, I'll talk a little bit about our work in looking at forever chemicals and their impacts on agriculture. So yeah, for this first part of the talk, just to motivate this is really thinking about a One Health approach to overcoming antimicrobial resistance. So this is the idea that um, human health is, inevitably tied to animal, planetary, and environmental health. And so, for this part of the talk, at least, emphasizing the fact that the use of antimicrobials anywhere, whether it's on a hospital or a farm, can contribute to antimicrobial resistance and therefore be challenging. So the use of antibiotics in agriculture can end up in land, water, ultimately in us, and be a major driver of antimicrobial resistance. And so I'll talk a little bit about that in this first part of the talk. So with regards to resistance, we focus a lot on biofilms as one of many modes of resistance. So, what is a biofilm? A biofilm is a mixture of DNA, proteins, lipids, and polysaccharides that, microbial cells secrete. This cartoon down here on the left kind of illustrates that process where a bacteria might attach to a surface. Once it attaches to a surface, it secretes these DNA, proteins, lipids, polysaccharides to encapsulate themselves and form a biofilm that can then grow, differentiate, and then be used to ultimately propagate um, throughout the environment, or in this case, in an implant or infection. So this encapsulation protects it, whether it's mechanical stress, thermal stress, environmental stress, or chemical cleaning methods, and therefore is a major driver of foodborne illness as well as crops and product loss. And that graphic on the right is a plate, an agar slant of a bacteria, Erwinia amylavra, that I'll talk about today. That is a prolific biofilm former and is the causative agent of fire blight. So polysaccharides are the major kind of chemical component present in biofilm. So, these polysaccharides play a number of very important roles in the microbial cell wall. There's a broad array of linkages and structures produced by different microorganisms and pathogens. And so that chemical structure really is important for the structure and architecture of biofilm, as well as its resistance and persistence, whether it's at various temperatures or in low water activity environments. So that graphic all the way on the left showing flagella, glycoproteins, fimbria. These are all examples of cell structures where polysaccharides play a very important role, whether it's cell motility, whether it's adhesion. Some of these structures, such as cellulose, PNAD, cholic acid, might be familiar to you. And on the right is kind of how these work in concert to encapsulate Erwinia, which is very important for its virulence. So I'll talk a lot about apples. So I wanted to spend a moment just to pay homage to the apple. Um, Apples are, it's a $32 billion market in the US, $99 billion globally. US is the second largest global producer and exporter. Where we're located in central Virginia. Virginia is a major apple producing state. You can see on this graphic from the USDA here. Um, Virginia is ranked among the highest. So it's really that kind of region between West Virginia, Virginia, Maryland, Western Maryland, and Pennsylvania. That's a major apple producing region. It's the number one consumed fruit in the U.S. Over 11 billion pounds produced. About two-thirds goes to food or consumption, about a third to juice products. So it's a major economic and agricultural driver in the U.S. And Erwinia amylavra, EA for short, or fire blight, is a major cause of apple and other crops such as pear loss. So fire blight is kind of a common term to describe it. It's caused by this pathogen, Erwinia amylavra, or EA. As you can see in these images here, it produces an exopolysaccharide slime or biofilm that encapsulates it and protects it from pesticides or treatments. There really isn't effective treatments that are uniformly effective in producing or preventing or reducing this infection. So as a result, agricultural antibiotics are often applied directly to trees to manage disease. And so this spread and severity is increasing due to climate change. So on the left, you can see a fruit surface with this ooze appearing. That's buildup of the biofilm as the pathogen is kind of spread. And on the right is a canker at the base of a tree where you see this ooze coming out, that is, another manifestation of this biofilm as this pathogen is uh, infected a tree. I'll talk a lot today about shoot blight and blossom blight. Those are part of the life cycles of the disease, and those happen in the spring. So this graphic kind of illustrates the life cycle. I think the important thing to take away here is kind of if you look at number 3 and number 4, that's bloom stage or king bloom. So that's when the flowers bloom. That's when the plant is often most vulnerable to infection, or one of the times that it's most vulnerable to infection, whether it's transmission by a insect or just rain, wind, or weather conditions brings the pathogen to the surface. And then once it's infected the tree, this shoot blight and blossom blight are kind of the first stages that you see at the beginning or onset. And so that's, the cankers that I showed are kind of late stage, summer, fall, and those allow it to overwinter and then propagate next year. Um, so there's a number of challenges in addressing this disease. So, agricultural antibiotics such as Fireline are currently approved for use on apples, but this article from 2024 kind of illustrates one of the challenges, which is streptomycin, which is one antibiotic which is used, has since been banned for use in citrus. There's kind of other bacterial diseases that affect that crop. And so there's increasing scrutiny around the use of these antibiotics to manage disease and kind of a constant need for alternatives. Other products such as Bordeaux mixture can be really toxic and therefore have limited efficacy against biofilm. So there's really a need always for new challenges to kind of overcome this biofilm and mitigate this disease. This just kind of illustrates what the consequence of it. So this is from 2018. This was a fire blade outbreak with out at Oregon State University. This is one of the partner universities we work with. And you can see really the management here just boils down to kind of cutting back the trees where the disease is present, burning that biomass to sort of mitigate the spread. And so, in a lot of cases, it's just you're kind of assuming a loss every year due to the spread of the disease and kind of managing it, assuming a 20 to 30% loss of your orchard due to this disease. So, my lab has studied a lot of different enzymes that are involved in cell wall synthesis and polysaccharide processing. These are some papers that we've published on different enzymes and their applications in food and agriculture. And so one today, CAS, which is a glycosyl hydrolase, is one that I'll talk about. Our thought was, based on what we had learned for use of these in other food applications, could we use this or other related enzymes as a tool to eliminate biofilm and overcome antimicrobial resistance for this specific fire blight disease? So it's part of the GH28 or glycosyl hydrolase 28 family of beta helix proteins. On the far right-hand side is a three-dimensional structural representation of this protein. This family includes both carbohydrate processing enzymes from fungi, phages, as well as some bacteria. And so a lot of the papers I referenced talk a lot about the biochemistry of this family of enzymes and how they work. So they're a useful model for us to study how we can design enzymes to target these types of carbohydrate structures. So this is just kind of getting into the details now of the pathogen. So, as I showed you before, it produces a robust biofilm. On the left here is SEM images showing just up close what that looks like. You can kind of see these individual cells that are elongated, but really a pretty thick buildup of that biofilm exopolysaccharide encapsulating them. And then on the right is a agar slant. This is just a field isolate that was taken from an infection in an orchard. And, you don't see individual colonies there. You just see this kind of slime layer or glossy slime layer growing over the surface. That's because it's producing an overabundance of biofilm on King's bee media. So this is just kind of its natural or kind of constitutive state is to make as much of this exopolysaccharide as possible. So, for us, we really wanted to start by evaluating these enzymes, engineering these enzymes, and seeing whether we could do that. So we had to develop a method to characterize this biofilm specifically. So as you could see before, it's not hard for this thing to generate biofilm. So we grew large amounts of this. We developed an extraction procedure to specifically purify the polysaccharides away from the cells. discard these other components, and then really hone in on that as something that we could target related to how we might overcome its resistance. So the mode of action here would be one where, the enzyme attacks these polysaccharide linkages specifically, depolymerizes these, then produces monosaccharides. And if that's the case, we should see evidence of that biochemically, and then in different applications where if we break that, we break down the biofilm, that might be a possible way that we can overcome some of the resistance that this pathogen shows. So we repurposed MTBH assay. So this is an assay that measures, free aldehyde groups present. This kind of also relates to the mechanism by which the enzyme works. So if it's a hydrolytic enzyme, it should generate free aldehyde groups that are reactive, and reactive in this case gives rise to a color change, this blue indicator. So it's a pretty simple visual test that we can use as a first test to evaluate activity against different biofilm isolates as well as different enzymes to see whether we could develop something that works. And this is just some representative data from some of these isolates. So 273 and 1430 are two field isolates that we took from orchards that had active infections. We purified these exopolysaccharides or EPS from them, and then just looked as over a function of time to see what happened. And so you can imagine this is a pretty thick, viscous biofilm. And so it took time for this enzyme to, presumably diffuse into this biofilm and illicit activity. But, as it accumulated over the course of really days, we began to see significant degradation to, say a high buildup of these monosaccharide products. So this CAS enzyme that we had characterized seemed to be effective, at least initially, in breaking down these purified biofilm components in vitro. We then took samples. We work closely with the USDA. So Joe Capobianco is at the Eastern Regional Research Center, helped us to do a lot of the imaging to look at how this is affecting the cells directly. We treated the cells, in this case, for 1 hour at a higher concentration of a gram per liter. That's shown on the right. On the left is no treatment, and you can see that, after 24 hours, this enzyme, was effective at stripping off the majority of that biofilm from the cell surface. So, even though it may have taken several days to really build up a large amount of monosaccharide products in the bioassay, shorter contact times were enough to really begin to strip this biofilm away and expose the cells, which is kind of a good first step. We wanted to characterize this biofilm because it's not just a single polysaccharide. This is an environmental extract to figure out what the components were that were present, and then look to see what was affected by enzyme treatment. And so we worked with the Complex Carbohydrate Research Center at University of Georgia to first characterize what was present in these samples, and then also look to see what was reduced. And so when we found some interesting differences among the different isolates and susceptibilities. The 273, for example, we saw a pretty substantial reduction in these galactopyranacil residues. And so that red arrow downward indicates the loss of these linkages post-enzyme treatment as compared to the untreated control. And when we looked a little bit deeper into kind of the chemistry and the biology of how this pathogen works, this is from an older study where they had done linkage analysis and NMR to actually determine the structure of amylavrin. So that's part of the name by which Erwinia amylavra got its name. This is a unique polysaccharide that this pathogen produces specifically that contributes to virulence. And these two linkages in red were two of the ones that were obviously identified as significantly reduced in our linkage analysis. And so at first pass, it suggested that these might be ones that the enzyme is active against. And on the right is taken from a review article that kind of talks about the mechanism by which this pathogen works. So it makes two types of major polysaccharides, levin, which is in blue, and that kind of ligates or binds the cells together. And then the amylavrin, which is yellow, which then encapsulates these ligated cells together to make that sort of thick biofilm. So, at first pass, our results would be consistent with the enzyme really attacking that yellow layer that's encapsulating the cells. As we explored this further, we also started to find some other interesting effects. So as we increased the enzyme concentration and we started to look across different isolates, we were able to see that, in many instances, as we continued to increase the enzyme concentration, we also began to see bacteriostatic effects using a CFU count assay. So in other words, the enzyme also exhibited some partial antimicrobial activity in addition to biofilm removal activity. And so we could, you can see here about a 1 log reduction. Depending on the isolate, the conditions used, we could go anywhere as high as, 1 to 2 log reduction. So that was interesting to us because that's a completely, that would seem to be something completely independent of the biofilm formation and the SEM that I showed you before. So it seemed as though, the linkages we found related to amylavrin are clearly ones that it's active against, but it may have other activities against other cell wall structures as well. So, given that result, we went back to Joe. We did some more electron microscopy against some of these other isolates. Then we started to find a lot of other interesting outcomes. So on the left here is an individual EA cell. You can see its sort of flagella present there. And on the right is a cell that's been treated with enzyme for 18 hours in solution at a concentration of 1 gram per liter. And you can see, this cell is significantly distorted, cell wall is disrupted, components sort of lost. So this would be consistent with that antimicrobial activity that we saw before. And then the facility that we have at the University of Virginia also allows us to do tomography. So we actually looked at sort of cross sections through the cell to try and understand where it might be active. And so on the left is just a cross section of the cell, no treatment. You can see it's a gram-negative bacteria. You can see the inner and outer membrane, as well as the internal structures of the cell. Whereas on the right, you can see this membrane is clearly distorted, right? And so the contents have sort of been leaking out of the cell. And so this again was kind of consistent with this direct antimicrobial activity that we saw as we approached higher concentrations for a lot of the isolates we were looking at. And then, to kind of revisit this before, as I mentioned, this yellow amylavrin polysaccharide is really important for virulence. And on the right kind of illustrates from this, from George Sundin's group at Michigan State, kind of a review article that talks about, mechanisms of action. So on part A, you have these individual cells, which are infecting leaf tissue in this case. They can interdigitate between individual cells. They can secrete toxins, which cause these cells to die. They release nutrients to allow the pathogen to grow, and then they begin to secrete this amylavrin. And that amylavrin kind of acts as a encapsulating agent to create sort of sliding motility that allows these things to slide through or migrate more easily through this tissue to disseminate and cause infection. So one way to think about this amylavrin is it's sort of a key step in the dissemination of it once it infects tissue. So we wanted to look at that aspect specifically. And interestingly, we used a soft agar motility assay. So this is an assay where we put the cells as it sounds in soft agar and look to see how they diffuse or spread. And so interestingly, at lower concentrations below the threshold where we saw cell membrane disruption, we saw a significant decrease in motility. And so it suggests then, that, consistent with what we thought in terms of how it was acting on the amylavrin, this would be consistent with that result, and suggesting that there's probably multiple modes of action that this enzyme's capable of producing. We wanted to also test this, outside of the lab and begin to investigate whether this is actually effective in a field application. And so over the past 3 years, we've been doing field applications with a number of academic partners in Oregon, Washington State, and Virginia. These are all major apple producing regions in the United States. And so in these field trial results, I'll show these trees have been infected with EA at King's bloom, or 90% King's bloom, at a 10 to the 7th CFU per mil concentration. And then after that disease pressure is built up, our product as well as others are applied to see what the reduction in infection is. And then, just to kind of normalize the language a little bit, percent disease incidence is just looking at the blighted flower clusters over the total population, and then the percent reduction is just the comparison between untreated and treated on a percentage basis. So these were some results from our trials. So Sergeant Akimovic is a professor at Virginia Tech that we work with, who's an expert in fire blight biology. and looking specifically at blossoms and shoot blight. And so, here what you can see is that as compared to an untreated control, there was a significant reduction with the enzyme at, applied once or twice. Um, Firewall and Fireline with Regulate, those are antibiotic controls. So it's significant relative control, and it's significant approaching the efficacy of antibiotic treatments. And so we're now continuing this trial for this year. We haven't wrapped up yet. We're doing our analysis, but this is sort of a promising series of results over multiple years showing that the enzyme's effective when directly sprayed onto the tree, the same way that you'd apply any other product. This was some results from recent trials in Washington State. So here we were comparing the enzyme to another biocontrol agent, Blossom Protect, as well as the streptomycin, which is a antibiotic control. Um, it kind of, as we saw in Virginia, the enzyme is significant relative to control, so it's active, and in this case has comparable activity to the biocontrol agent Blossom Protect. So again, kind of a good indication that in a completely different climate, environment, and application method, consistent results. And then what we were also interested in is looking at whether we could integrate this into a IPM or management strategy that allows us to mitigate or reduce the amount of antibiotics used. So um, we've begun to combine this with all sorts of other applications. And so what this shows is that by adding the enzyme in combination with antibiotic, we can reduce the amount of antibiotic used to achieve significant reduction, equivalent to that of just antibiotic alone. And so this is useful because this might be one potential strategy that could be used in the future to help reduce the use of antibiotics in this application. And then just to mention briefly that this is academic research, but there's a translational component to it. As mentioned before, I founded this company, Lactose Technologies, to help commercialize the use of this and other applications in food and agriculture with really the goal of trying to remove, overcome antimicrobial resistance and find more sustainable solutions to challenges such as these. So to summarize this part of the talk then, I think our initial results indicate that this enzyme is effective in degrading purified biofilms in vitro, and it can directly remove biofilm from cells. That specificity that it shows is most consistent with amyloidin degradation, and that was also shown in these motility assays, which is a key component of the biofilm. At higher concentrations also can damage cell walls, so kind of a mixed mode of action, and that in direct spray applications, it was effective, and in some cases comparable to antibiotics or biocontrol. So it might hold some promise for use eventually in actual commercial applications. And I just want to make sure I acknowledge Kevin, who is the graduate student working on this project in my lab, Cole and Evan Culp, who are two undergraduates who've worked on this, Surgeon at Virginia Tech, who's leading this USDA grant that we're a part of that's funded the work, and then Frank and Joe, who are key collaborators that really helped us expand our testing and understanding the mechanism of how this works. Great. So now I wanted to switch gears and talk about a different aspect of the work that we're doing. And this is really looking at detecting and removing forever chemicals in agriculture. So I'd say thematically, this kind of fits under the umbrella of what I call or what is referred to as the exposome. So this is the idea that exposure over a person's lifetime to environmental contaminants or various environmental factors can really contribute significantly to their health, overall health. And there are many different graphics you can find that illustrates this. I like this one because it kind of shows it in a timeline format, right, or a cumulative format. So and the link down there is to an article from the NIH that recently this summer NIH announced a global exposome initiative to really begin to look at the linkage between chemical exposure, human health, such as illustrated here, right? So 80% of human diseases are associated with environmental factors and chronic exposure, unique to each individual, and beginning to understand that as something that is really important for both human health as well as environmental health. And then so we work very closely with colleagues in Maine at the University of Maine. And I like to show this picture because it kind of just illustrates one of the big challenges in working on a problem such as PFAS or forever chemicals. So this is from our summer field testing. So this is a Malabeam Lake in Aroostook County. Yeah, it's a beautiful location, as you can see in the picture. But the water levels of PFAS at that time were over 100 parts per trillion. So, hundreds, if not thousands of times higher than the current EPA accepted limits for drinking water. So that's kind of one of the major challenges, right? Is that it's kind of ubiquitous in the environment. It's, increasingly present everywhere we are, but it's not obvious from looking at the environment where it is or what kind of exposure you're getting to it. And so that's one of the big challenges in addressing this. This is from December 2024. This is kind of the draft list, that human health criteria for three of the key PFAS compounds. This is in water. And what I would point out here is, the exceedingly low level for PFOA and PFOS, so sub, parts per trillion. So, essentially, exceedingly low levels for limits of detection and exposure. So as I mentioned, they're everywhere, right? And there's over 4,000 known compounds. They're ubiquitous in food, consumer products, industrial processing. They take over 1,000 years to fully biodegrade in the environment, hence the name forever. And there are specific compounds within the PFAS family that have been linked to health issues. That's shown on the right here, whether it's developmental issues, thyroid issues, cancer, immunosuppressant, many different studies, and this is a constantly evolving field as we learn more about what these are. And so on the right is just one of many graphics you'll find that kind of talk about where they show up. Because of their unique water repellent properties, they're very popular in everything from nonstick cookware, fast food packaging, paints, pesticides. Their flame retardant properties make them particularly popular in firefighting foams, and so historically, that's why they've been so prevalent in so many different consumer products in our everyday life. In agriculture, then there's a number of challenges in terms of tracing how they end up in the food we eat, and so it's thought that food and water are two major routes of exposure that people have for exposure to PFAS compounds. And so this graphic kind of illustrates that, whether it's biosolids, which can accumulate these materials from wastewater, whether it's irrigation water, these all end up in soil. People can come directly in contact with that soil. That can end up accumulating in crops, which we then eat or animals eat. The manure from the animals then spreads it and propagates it back into the soil, all of which ultimately leads into the food that we consume. And so, it's thought that about a third of agrochemicals, such as pesticides, are fluorinated to increase their stability. And fluorination is also common in food consumer products and chemical packaging to reduce oxygen and moisture. And so, the link down here is from an EPA study. Byproducts of fluorination processes such as these include the formation of PFAS compounds, and these compounds can then leach into food, pesticides, and other packaged materials. So even at a very small level, this byproduct can be significant in terms of ending up in food, the environment, and ultimately our exposure. So we work closely with growers directly up in Maine as part of this project. So Randy, who's in the picture here, he's director of the Central Aroostook Soil and Water Conservation District. He's a farmer. He's also teaches at University of Maine. And so, what I'm showing here is a picture from his farm. And so you can see on the right, he has things labeled PFAS broccoli, PFAS Brussels sprouts. That's because through testing, the irrigation water that he used from the Aroostook River, had significant levels of PFAS. And so this is kind of a challenge that a lot of growers face today is knowing that this problem exists and that it's ending up in the food supply. How do we address it? And so, a big part of our work and what we're trying to do is develop tools to help them identify previously unknown or uncontrolled sources of PFAS to kind of mitigate exposure, as well as understanding the fate and transport of these molecules in the food supply. Another challenge that kind of that we became aware of as we started to work on this was just the challenge to do rapid testing, right? So if you imagine a situation like this, you'd like to be able to go rapidly perhaps multiple times a day, and test at various points in the field, your crops, your water, your pesticide formulations to kind of figure out in real time whether you have an issue or not. And so, currently LC-MS is kind of the gold standard. It's primarily limited to water and wastewater samples. There are new methods now being developed for other types of solid matrices. The cost is high, and time can also be a challenge too. So typically, you're talking hundreds of dollars, multiple days per test. And so, that and LCMS is gives you a really rich data set in terms of chemical information and very detailed information about what's in the materials. But, this can be challenging if you want to be able to sample rapidly at multiple points in your field multiple times a day for multiple crops. So there's really a need for these kind of low cost, high throughput testing methods. And so that's kind of where our work really came in is, and these are some of the earlier publications from our lab where we developed kind of low cost, high throughput biosensors. So I'll show some results in the upcoming slides where we can use these directly on different types of field samples without needing pre-processing. We can detect major compounds like PFOA and PFOS. Um, it's rapid and relatively low cost. And so, this isn't meant to replace any other methods. I think the reality is we're going to need lots of different types of testing methods with lots of different levels of sensitivity to really achieve comprehensively what's needed to understand the fate and transport of these. But we see what we're doing as kind of one of many useful methods that allow us to get rapid answers to really flag something as a problem to then go on and do additional study. So as a our lab does a lot in the area of sort of protein engineering, structural biology, and we became really interested in the use of liver fatty acid binding protein, or LFABP for short, as a protein scaffold. So this is a protein that's present in the human body. Its expression is upregulated in response to PFOA, and it's known to bind compounds such as PFOA and PFOS with moderate affinity. And so the structure that I'm showing you here is taken from a crystal structure bound with a fatty acid, palmitic acid. And so the chemical structure is drawn below, and on the, just to the right of that are two of the major PFAS breakdown products in the environment, PFOA, PFOS. So not identical, chain links are quite different. One's fluorinated, one's not. But nonetheless, as a first pass, we thought this could be an interesting scaffold to work off of to design more effective binding elements to turn into biosensors. And so we created two different outputs, one using a solvochromatic fluorophore, and then more recently a genetically encoded version that uses GFP. So I'll talk briefly about both of those now. So first, the solvochromatic fluorophores. The idea here is that we did sort of a series of modeling and mutagenic studies to find hotspots within the binding site that were highly sensitive to different PFAS compounds. And then based on those compounds, introducing a fluorophore, in this case, acrylidin, whose fluorescence is highly sensitive to the environment in which it's exposed to. So, for example, a PFAS-free versus a PFAS-bound environment causes a shift in fluorescence, which is detectable. And so that's what you're seeing in that graph on below, is that X represents, as you can see, the shift, in spectra as we titrate in PFOA in one of our early versions of this. And so using this approach, we were able to develop these, kind of binding proteins or kind of proto-biosensors, from which we could use acrylidin as a probe to study binding. And you can see here very high limits of detection in the hundreds of parts per billion. This is quite high, certainly much higher than is necessary for water, but it's kind of our early work in trying to refine and build a prototype for this. We then wanted to look at just, how interference would happen, rather than using a neat laboratory buffer, say in a surface water sample, there's going to be a lot of other organic material present, or surfactants or other types of compounds, how does this respond when those are present? SDS is sort of a surfactant that binds this fatty acid binding protein with higher affinity. We're able to show that even the presence of equivalent or higher amounts of surfactant, that we're still able to get significant signal for both PFOA and PFOS. Then we wanted to test whether this could actually be used in some of the field applications. I showed before, like Malabine Lake, where we had hundreds of ppb levels, and so we worked closely with the Aroostook Band of the Micmac Nation. So the graphic there on the right is Maine. That orange star is where Loring Air Force Base is located. This is a known site where there has been a lot of PFAS contamination from aqueous firefighting foams. And so working with the Micmac Nation, who now have much of the lands there, we sought out to do some testing and see whether these sensors could be useful so that some of the land that was contaminated, we could figure out what to do with it or to determine ways that we might be able to ultimately remediate it for use to bring it back to agricultural uses again. So it's got a little bit of like a detective hunt. So, first is like, where are the sources of PFAS? So we went around to different water sources that are commonly used for irrigation, collected samples, took these samples, and that's what's shown on the bottom of the right-hand side there were just some representative samples, tested these with the biosensor. And so that shift downward you can see is reflected on the graph in the lower left-hand corner. So that reduction you see is indicative of binding relative to untreated control. So, we use this as almost kind of a yes, no when we started thinking about how we might use this, just to indicate whether there's a high level of PFAS present or not in these samples. We're using PFOA as a standard above, but this really isn't telling us anything specifically about what's in there, just that there's something present. We then expanded this to look at other types of samples, throughout Aroostook County, some soil samples, other water samples from various locations, some of which were kind of more heavily contaminated where we had to wear protective equipment. And then we did a follow-up study that was published last year where we wanted to do a head-on comparison with our sensor versus LC-MS-MS. And so this is just some results summarized from that. So if we took a summation of the verified levels, in this case of PFOA, and we compared the calculated, verified using the biosensor and the verified using LC-MS-MS, in a lot of cases we actually saw pretty good agreement between the two. And so Chapman Pit, Malabine Lake, these are some specific locations in Aroostook County we tested. And so this was encouraging to us because it suggested that, even in the presence of a natural background, say in surface water samples, we're still able to show enough sensitivity and specificity that we get good agreement with LC-MS-MS results. And then more recently, we wanted to explore how we might expand this or modify this or create other types of biosensors for other uses. And so we looked at genetic engineering tools to try and make, in this case, a genetically encoded biosensor, which could maybe be used in cell-based or in vivo detection. So here, what we were interested in was integrating green fluorescent protein, or GFP, into this biosensor in some way that the fluorescent signal is sensitive to binding of PFAS. The idea being that in the bound state, there's an increase in fluorescence, and that gives us sort of a direct readout, independent of needing to label the protein exogenously like we did with the acrylidin. And so we used a circular permutation approach in which we found permissive sites present in both GFP and the binding domain, screened a series of these different combinations, and then created ones where we changed the microenvironment around the chromophore to create a new fusion protein, which can then refold upon binding of a ligand, in this case, PFOA or PFOS. And so through this iterative process, we came up with these different permuted forms. The one shown here is the one that seemed to work the best in our hands and gave kind of the requisite outcome, which is upon ligand binding, there's an increase in fluorescence. And this shows here just titrations with increasing amounts of PFOA. And you can see this shift in emission spectra, increase in emission spectra after excitation at 485. So this is the excitation emission wavelength for GFP. And then if we converted that into a titration or kind of single site binding isotherm, we could see pretty good binding isotherms for single site binding, whether it's in PBS laboratory buffer, whether it's in a spiked creek water sample, or laboratory buffer PBS that contained an interfering substance like SDS. So Yeah, it suggested that proof of concept wise, we could use this as a as an eventual genetically encoded biosensor um, to expand the ways in which we can test for these compounds. And so the split protein seemed to retain a similar binding affinity to the fully folded. And then depending on how we transduce the signal, can potentially get down to perhaps even parts per billion or subparts per billion range um, without additional labeling. And this is kind of just looking ahead. I talked a lot about water and soil, but Maine has really led the way a lot in a lot of ways in terms of our knowledge around PFAS, fate, and its impacts in agriculture. And so the Maine CDC, for example, sets action levels for PFOA and PFOS and milk and beef. They're in the hundreds of parts per trillion or even parts per billion. So yeah, I would just point out that's several orders of magnitude larger than limits that the EPA has set for water. And so we've begun to look at how we might use these sensors to expand our testing into other types of matrices, such as meat. And so this is preliminary data, but what it's showing is that we can see a significant fractional change, that kind of single site binding isotherm in a diluted sample containing a beef extract that's been spiked with, in this case, PFOA. And so these are early results. We obviously need to develop this further, but it suggests that this might be a platform we could expand, or one potential application could be looking at other types of food matrices, food products, um, using this approach. Another thing I'll mention is, we're also trying to understand the transport of these things throughout the environment and their accumulation in crops. So potatoes are a major food product produced in Aroostook County. And this is a study that we did with Randy in 2024. And this is a report that he put out showing, accumulation of different compounds, whether it was in roots, leaves, or tubers from different irrigation sites, which were known to be contaminated. And so what was interesting to us was the kind of the biodistribution of these things. So in this case, primarily in the vegetative tissue, but not in the edible portions of the plant. And so this is another aspect of the work we're trying to achieve, is how can we figure out strategies to figure out where it accumulates in plants, perhaps mitigate that, with the goal of figuring out whether there's ways to augment it so that it doesn't bioaccumulate in edible parts of crops, or in this case, in the case of the vegetative tissue, that's not plowed back under into the soil where it might continue to propagate. And I'll just mention briefly, we're also looking at other types of remediation approaches, whether it's industrial hemp or other crop types that might be useful at bioaccumulating these things out of soil. So to conclude then, I think we showed some results that indicate these biosensor strategies are effective in detecting key compounds at high but relevant concentrations that were found in surface and groundwater. We can use this potentially for detection of other matrices, such as meat or complex matrices. These future strategies are underway to improve specificity and limit of detection, so we can try to build upon this rapid testing. And so Mahabub is a graduate student in my lab at UVA who's working on this, and Hai and Mike are partners in biology who've been kind of essential to all the work that we're doing, particularly on the genetic engineering side. And so a number of partners to thank, both industry and government, particularly the EPA, for supporting this work. So with that, I'll conclude and happy to answer any questions.
Okay, yeah, very interesting. I have a couple, but I think that we'll start with audience questions first. And then I'll remind our audience you can type in your questions through the Zoom Q&A feature, or you can raise your hand and I can unmute you to ask live. So to start us off, our first question is your biosensor for PFAS detection recyclable to reuse? And how did you concentrate your sample solution to meet such high, to meet, let me just start over. How did you concentrate your sample solution to meet such a high ppb or ppm of LOD level?
Yeah, fantastic question. So the first part of your question was around the recyclability of it.
Yes. Is your biosensor for PFAS detection recyclable to reuse?
Yeah, that's a great question. We haven't looked at it in that format, but that actually was a thought that came up and kind of motivated us to think about a genetically encoded biosensor, because if a cell is continuously producing it, there would be a way to, continuously regenerate it. So I guess that's sort of the best strategy we came up with to date. On the high concentration side, most of the work I showed with the high concentrations were either environmental samples where that high concentration was already present, or we prepared in a laboratory at that high concentration. So we didn't do a lot of pre-concentrating or sample pre-processing to concentrate it prior to detection. But that's something that we could, potentially use as a tool to help use our biosensor in applications where the levels might be lower.
Great. Our next question is, what are the ways to improve the sensitivity of the biosensor to PPT concentrations?
Yeah, no, that's a great question. That's a lot of the work that we're doing at the moment. So a lot of it is really it's kind of multifaceted. We're working with partners who can help us improve the transduction of the signal downstream. So better, more efficient ways of capturing the fluorescence. What we had done to date was really just using a basic plate reader in the lab, which is not as sensitive as other types of fluorescence detection methods. We're also looking at other types of proteins that might be able to transduce a signal more sensitively than GFP. And then doing a lot of redesign of the binding site using sort of protein engineering techniques to improve that KD or that binding affinity. Because palmitic acid is much longer, the PFOA is much smaller, so redesigning that binding pocket to make it more specific and create higher affinity is something that we're working on as well.
Okay, maybe a couple of my questions. So, thinking about the enzyme used to reduce the biofilm on the apple trees,
Yeah,
could that be applied to other fields? Like, so I'm thinking specifically like Legionella that produces the biofilms and then is hard to treat and has
yeah.
all kinds of other problems. So can we translate it to other areas?
No, yeah, no, that's a great question. In fact, yeah, some of the work that I referred to earlier works more on like human pathogens, uh,
Okay.
and medical and other settings. So yes, that's something that we've talked about primarily more on the commercialization side. And so I think that that's something that this could be very useful for.
Okay. Another question that came in, how does the selectivity of your sensor in the mixture of solution of PFAS?
Oh, if you have like a mixture of different
Yeah,
PFAS
yeah.
compounds. Yeah, we haven't really been able to achieve great selectivity for a specific compound yet. So I would say it's pretty permissive. I think we've tested things as short. There is some selectivity the shorter the chain. Like if you look at Gen X or short chain PFAS compounds, it tends to not be sensitive to those. I think we tested a PFHxS, 6 chain um, compound, and it was sensitive to that. But I think for now, I really kind of think of it as just sort of a broad spectrum or broad brush biosensor for the presence of it or not versus specifically to PFOA, PFOS. In the papers that we published that I referred to, we did see some preference for PFOA over PFOS. There's some kind of intrinsic selectivity, but um, I kind of think of it more as just a broad sensor as opposed to something specific for a particular compound.
One of my other questions, so I kind of do like research, translation, and outreach type things. What are like people's sentiments as far as like testing for contaminants and PFAS in like various fields? We've seen the spectrum from like I don't want to know what's there versus like I want to know what's there and how to fix it and that kind of stuff. So,
Yeah, no, that's a great question, and that's something that I think we faced also when we wanted to start on this project. So, yeah, I think we had a very similar experience to what you described. We're kind of ran the gamut from folks that were very, very eager to understand, and then others who, had concerns. And so, I think for us, it's always kind of approaching people from the standpoint of like sharing, information sharing, sharing new knowledge together, kind of working collaboratively in close communication. I'd say the Micmac Nation is a great partner to work with. They're really like a great, just a really like scientifically curious group of people that are just really passionate about solving these problems and then sharing that knowledge with others. Um, so we were very fortunate to find them and very fortunate to work with them. Um, yeah.
We don't have any more questions come in. So maybe if you want to give any final thoughts or advice for the next generation of scientists.
Oh, well, yeah, I mean, I hopefully the talk today kind of illustrated some of the big like challenges that we face, whether it's environmental antimicrobial resistance or these contaminants of concern. I think they're important areas that are going to take lots of different diverse teams to work together. Yeah, I'm trained as a chemical engineer. I graduated from School of Chemical Sciences at University of Illinois. But yeah, I think being open and willing to kind of take the challenge on and kind of look at it unconventionally to try and come up with new solutions is going to be really necessary. And that's going to mean lots of different backgrounds, educational experiences, and an interest to be successful. I think these are really, 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 truly collaborative problems just because of how big they are. Um, so that would be my advice.
Yeah, yeah, wonderful. Thank you. So thank you again for joining us, and we'll conclude the webinar.
Thanks. Thanks for the opportunity.